All right, so here we're going to talk about glycogen use in muscle cells. So, muscle cells will have glycogen stores, and the glycogen stores are a very important source to tap into energy as needed. They are going to have differences in regulation with respect to the liver, and some of this is mostly because of the hormones. So, glucagon will never affect the muscle cell. Glucagon can affect the liver cell and tell the liver cell that we are now in the fasting state, but the muscle doesn't get affected by glucagon. Glucagon is a mobilizer of glycogen in liver. Now, what does have an effect on the muscle cell is need. So, if you're exercising or if you have an increase in stress, your fight or flight, then you're going to get activation of your glycogen phosphorylase, and this is going to cause degradation of glycogen stores. AMP is an allosteric activator of the glycogen phosphorylase in muscles. In addition, calcium in muscles from release from the sarcoplasmic reticulum is a stimulator. Glucose is not a physiologic inhibitor of glycogen phosphorylase A in the muscle cells, and glycogen is a strong feedback inhibitor of muscle glycogen synthase. Epinephrine is going to stimulate phosphorylation by pKa, and then you're going to have stimulation and degradation are similar both in the muscle and the liver cell with respect to epinephrine. All right, so exercise, of course, is going to activate the mobilization of energy stores such as glycogen. So exercise is going to activate glycogenolysis. You're going to have muscle contraction, nerve impulses, or epinephrine, a use up of ATP, an increase of AMP, and all of these are going to have the effect. Now keep in mind in the liver, your glycogen is going to lead to a release of glucose to maintain blood glucose levels. In the muscle, your glycogen stores are going to be used for energy. So it's going to convert it to glucose 1-phosphate to glucose 6-phosphate and enter glycolysis. Now there are two ways to activate your muscle glycogen phosphorylase B. So muscle glycogen phosphorylase B is not active. It can get phosphorylated by a phosphorylase kinase, and this will add phosphate groups and open up muscle glycogen phosphorylase B to A. Now it's active. Alternatively, AMP has another binding site, so the phosphate will have one binding site, AMP will have its own allosteric binding site. Activation of muscle glycogen phosphorylase will occur with AMP binding to its allosteric sites, and again opening up. And so these substrate sites are now open, so you can have your glycosyl units bind at the non-reducing ends, and the glycogen molecules will be degraded. Now, phosphorylase kinase is going to transfer phosphates from ATP to serine residues in each subunit of your muscle glycogen phosphorylase B. Now, so basically, we still have those two ways to activate your glycogen phosphorylase B. You can either activate it by adding a phosphate, or you can activate it by binding to AMP. Both of them will have the same effect. Now, the activation of your phosphorylase kinase can happen in two different ways. You can have a nerve impulse, which triggers calcium release, and the calcium calmodulin will activate phosphorylase kinase. You can also have hormone, the fight or flight, epinephrine, that will activate cyclic AMP, protein kinase A, which then can activate phosphorylase kinase. Phosphorylase kinase is activated by a phosphate, and phosphorylase kinase will, in addition, will then phosphorylate glycogen phosphorylase B to form glycogen phosphorylase A. So in essence, glucagon, epinephrine are going to lead to the release of glycogen, lead to phosphorylation of glycogen synthase and activating it, and glycogen phosphorylase activating it. This is going to occur both in the liver and in the muscle. This is only epinephrine. Muscle glycogen phosphorylase is allosterically activated by AMP and by increases in calcium levels, um, through synapses or through epinephrine fight or flight.